So the idea why we are going for this, we're doing all this elaborate mechanism, is trying to find out why does cancer relapse after many years of apparent remission. You give somebody chemotherapy, you think the cancer is in remission. You must have heard this term a number of times in patients with cancer. It was in remission, now it has relapsed. Now, if the surgery, and especially in an organ like breast, which can be removed in total, why does after removing the cancer in total, does the patient get a relapse? The patient is getting relapsed because there were metastases which had happened, which are not detectable. So we are trying to find out ways and means by which those populations can be identified and we can target them. Okay, this is just a slide, very busy slide, but please try to understand that what we are trying to observe here is that the cells came from a mouse transgenic model and a rat adenocarcinoma model. But this is like the cell model and this is like the genetic model. Both of them show that the cancer cells that are in the invasive cells are hypoapoptotic. So you challenge them with chemotherapy drug, they're not going to die. So now here's a big problem. The problem is these cells you cannot detect them, you cannot kill them, or they might kill you. So our job is to try and find out ways by which we can kill those cells which might kill you. So here we are. We observed, this is again a little busy slide, but I want to tell you that we are looking at the 4 populations that I described a few minutes back: the average primary tumor cells, the invasive tumor cells, the circulating tumor cells, and the metastatic cells. If you give them a chemotherapy, tamoxifen, very common chemotherapy agent, and doxorubicin, still the average primary tumor cells die, the lung mets die, but the cells in transit live. So this is exactly the problem. If you give chemotherapy after surgery, whatever primary tumor is left over is going to die by the chemotherapy. Whatever successful lung met, big lung met has formed, that is going to die, but the cells in transit are not going to die. Just to remind you where we took all these 4 populations from, we took the average primary tumor cell from the tumor. The invasive tumor cells were collected by the in vivo invasion assay that I just showed you. The needle collection, where the cells, think they're getting into a blood vessel, but they're actually moving into our needle. Cells that have successfully entered blood, and cells that have gone on to make lung metastasis. Okay? So here what I'm showing you is that if you give chemotherapy to the population, all the 4 different populations, only the ones that are in the tumor or have already formed large mets are going to die. So probably now we have a population which is the drug-resistant population and is the metastatic population. Okay, now how do we target it? Okay, we have identified it, how do we target it? This is a slide just to show that exactly the same thing happens when you give radiation also. So if you thought that if you're resistant to chemotherapy, radiation therapy is going to cure you, no, that's not going to happen because these cells are radio-resistant as well. Same idea, average primary tumor cell, invasive cells, circulating tumor cells, and metastatic tumor cells. So the drugs are going to kill, the radiation is going to kill the average primary tumor cells and the metastatic cells. The cells in transit will continue to live. Now, since I told you that we have the capability of looking at the behavior of the invasive cell, both inside the animal as well as outside ex vivo, we designed this assay. We were not the first to design this assay. This assay was published by other people, and we just utilized it. It's an assay called COMET assay. Basically what it does is you let the cells grow for about 16 hours after you have taken them out of the animal, and you give some gamma radiation that breaks the DNA, and then you let the cells repair their DNA for 1 hour. After that, you put it in a electrophoresis plate and electrophorese the cells. Those of you who are paying attention, DNA repair pathway was actually upregulated in the invasive cells. In the pie chart that I showed you, one of the pathways that was upregulated was DNA repair pathway. Now here is our mechanism, here is our experiment to test that phenomenon. Okay, the idea that we got from the gene expression studies was a bioinformatic one. Yes, some of the genes are upregulated, but is that biologically true also? In order to do that, we did this experiment And I'll just show you one slide. So basically this assay is very simple and very easy to do. So Dashan's son, my postdoc, just set it up in one day, and within like a week she had all the data. So it was fabulous. It rarely happens when an assay works so easily. So if you see this is the nucleus of a cell. This is the nucleus, this is the nucleus, that's the nucleus. And what we are doing is, we are running electrophoresis in this site, okay? So if you have large DNA particles, they're going to stay back inside the nuclear zone. If you have degraded DNA or DNA that has gotten broken down, or in this case, not repaired, it's going to move this way, okay? Then we find out how much DNA we have left in here and compare that to this tail DNA, as we like to call it. And it looks like a comet, that's why they call it a comet assay. Okay? So using this analysis, what we were able to do, and unfortunately this is not a direct representation of my screen, this actually happens to be the average primary tumor cells. So you can see there's a big population which is not able to repair its DNA after you give it 3-day radiation. So why? Because it's not able to repair its DNA, it's going to die. Whereas the invasive cells, with the same amount of radiation, they're able to repair their DNA. This is percentage of DNA in the tail. So if you have 100% of the DNA in the tail, that means everything is broken. This cell is not going to survive. Whereas if you have like 10% of the cells in the DNA, then the cell is able to repair. So using this assay, we found out by a biological an experiment that yes, the invasive cells are able to repair their DNA more. This is very exciting because remember I showed you a few minutes back that these are the cells that are able to fight off chemotherapy, fight off radiotherapy. So that's because chemotherapy and radiotherapy essentially cause the damage by damaging the DNA. So if you have the capability to repair it quickly, you will not get affected by the drug. Is it clear? What I'm just saying? No, obviously not. Okay, please try to understand. Here you have a chemotherapy or a radiotherapy which you are providing. It essentially targets rapidly proliferating cells because that's what cancer cells are. And then, you observe that there's a population that is not proliferating and yet has a very high capability of repairing their DNA. So now if you give them chemotherapy, that population is not going to die. That is going to remain dormant for many, many years and come back as a relapse. Okay? So we believe that here we have a method for identifying those cells and trying to target them. I mean, I haven't come to the targeting part as yet, but we are still identifying them. Okay, so enough about introduction, enough about understanding. So basically what I can say is that the patterns of gene expression and biological properties unique to the invasive subpopulation of cells have been identified, and they are these, which I have been repeating so many times. They are neither proliferating nor apoptotic, but are highly motile and resistant to chemotherapy and radiotherapy by upregulating the DNA repair mechanisms. Alright, it's not a doomsday scenario. So what can we do about it? A couple of things. At least in my laboratory, we're doing 3 things. I will talk a little bit about the biomarkers. I'll just give you one example of a pathway-directed drug, and I'll give you one example of a radio or chemosensitive drug. So what are the things that we're doing? Like I said right at the beginning of the talk, the early detection is the key. You not only need to know that you have cancer, you need to know what kind of cancer you have. Is this cancer going to be a highly aggressive one? Is this cancer going to be a highly metastatic one? Or is it benign kind of a cancer which can be treated with less aggressive treatment? This is a decision that a clinician has to take very early on in the treatment. When, unfortunately, we still don't have a blood marker, we don't have a mechanism by which in your annual screening we can detect a tumor before it is big enough that it gets detected by a mammogram or something like that. Usually, even before that, the lady observes a lump and reports to the doctor. Alright? So what we're trying to work on is, if not at the blood level, but at least when the lady reports a lump, we should be able to do a fine needle aspiration or something like that to find out what is the quality of this, quality of this tumor. Is this going to be a very aggressive one? If yes, then we should go ahead with all the treatment that we have in our arsenal. Or is this going to be not so aggressive? As it turns out, more than, okay, I won't give you a number because I'm hearing contradictory numbers from two different sources, so I won't give you any numbers. But a big share of the chemotherapy that is going on in the patients is not needed. They will never have metastasis, whether we give them the chemotherapy or not. Now, those of you who have ever seen anybody go through chemotherapy know that if anybody can avoid it, there is no better way than that. Chemotherapy gives people lose hair, they become thin, or it's a terrible thing. So, if you can avoid that, all the more better. So we are trying to identify biomarkers that will actually tell the doctor right at the beginning of the treatment that this lady does not need chemotherapy or definitely needs chemotherapy. Okay? So, and the second research that we are doing in my laboratory is we are trying to identify drugs that would specifically kill the invasive and circulating tumor cells. The ones that I showed you a few minutes back would not die with classical chemotherapy or radiotherapy. And the other line of research that's going on in my lab is that we are trying to identify radio and chemosensitizers. I will show you only one today of each, which makes these so-called resistant cells sensitive. So now if you add to your chemotherapy regimen this chemosensitizer, you're going to get the desired result that you want. You will kill cells from all those four stages. The ones, the average primary tumor, the invasive cells, the circulating tumor cells, and the metastatic cells. Remember, the metastatic cells came from the lung, the circulating tumor cells came from the blood, and the invasive cells came from the assay that we do. And the average primary tumor cells came from the tumor that the patient started with. Okay. There are two areas of research which we do for the biomarker analysis. One is pathway analysis, another is splice variant analysis. Splice variants are, without getting into much complication details, they are different versions of the same gene which selectively get upregulated in some cancers, in the aggressive cancers, and not in the other. We have identified a few of those. In fact, one of them is already patented by Albert Einstein College of Medicine, and we are signing a contract on 26th with a company. Hopefully we'll go to the clinic. So this is the outcome of many years of research, at least 8 years on my part. Even before that, many people have done this. So it's not trivial to come up with something that will go into the clinic. So using this model, I just wanted to say one thing before I go on. I just want to remind you again that the pathways that we had identified by bioinformatic analysis, which were upregulated in the invasive cells, was the DNA repair and motility up, cell cycle, and apoptosis down. Because cell cycle and apoptosis is down, these cells don't get activated by they don't die out of the chemotherapy or the radiotherapy that you do to them. Because these are the mechanisms that are utilized by those drugs. And the DNA repair being upregulated also saves the cancer cell. Okay, so let's move on. Don't worry, I'm not going to go through this entire slide. I just want to point 2 points. One, look at this EGFR, epidermal growth factor receptor. Okay? And this protein called MENA. The red arrows in bracket tell you that in our bioinformatic analysis, we have found them to be upregulated. Wherever you see green colored bracket, it is downregulated. Wherever you see red colored bracket, it's upregulated. It's a very complex thing to do because not every patient is the same. Every patient has a different genetic background. So what you have to understand from this gene expression analysis is the underlying mechanism. And that's exactly what we are trying to do. So once we look at the motility pathway and see that the epidermal growth factor receptor is upregulated in both the models that we have, and MENA is upregulated in both the models, we decided to focus on those in a little more detail.